The enzymatic method for DNA sequencing is also known as Sanger sequencing, after its developer Frederick Sanger, who first described the method in 1977. The result of the enzymatic method is very similar to the chemical method, but the procedure is completely different. First step, polymerase chain reaction, PCR, with fluorescent chain-terminating dd-NTPs. First, a primer is transferred to 4 tubes. Then, DNA polymerase is added to each tube. Subsequently, dNTPs are added to each tube, and then specific dd-NTPs, dideoxynucleotides, which can be guanine, adenine, cytosine, or thymine, are put on each tube. Then, the tubes are collocated in the thermocycler, and an N-chain PCR is performed. Each tube has sequences that end with a specific nucleotide, depending on the dd-NTP added. Second step, size preparation by gel electrophoresis. On the next step, gel electrophoresis is performed, putting each sample containing different dd-NTPs in separate lanes. Due to the fact that DNA is negatively charged, the molecules will travel to the positively charged region, and the heavier DNA fragments will take longer to travel along the gel. Third step, gel analysis and determination of DNA sequence. It is important to label whether a primer or a dNTP with radioactive phosphorus, 32P, in order to be able to see the DNA bands in the X-ray film. Overnight, the DNA fragments will stick to the X-ray film, and the fragments will be visible in the sequence ready to read. Finally, it's worth mentioning that Sanger sequencing can be conducted whether in manual form or in an automated manner.
In 2015, some students from the Applied and Biomedical Sciences School in Australia did a practice through the search sequencing method, looking and creating their primers to be able to find the complete genome of influenza A virus, AIV, and determine if resorting has occurred or if any other pathogenic traits were present. However, after a long experimentation, they were not able to design a set of primers that could universally amplify the 6 internal genes from influenza A subtypes. Their experiment was functional to 3 subtype viruses that they chose, proving that search sequencing method was useful, but influenza A virus had too many variants for them to find all the primers needed for the sequencing.